Hello, everyone. If you are new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. Today, our presentation is how to establish the state-of-the-art within the Scientific Validity Report. Our presenter today is Dr. Sarah Chavez, Principal Medical Writer with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I will welcome Sarah to give an overview of our session today.
Okay, thank you, Olivia, and thank you to everyone who's joining us today. During today's session, we will be reviewing the scope and the purpose of the Scientific Validity Report, which I will refer to as the SVR. And in particular, we're going to be discussing how the SVR helps you to establish that your IVD is state-of-the-art. So really, our goal is that by the end of this presentation, you will all have a better understanding of, first off, the importance of planning and preparation before you begin to write the SVR. Secondly, what should be included within the SVR. Third, what is meant by a systematic literature review or an SLR. And finally, how to establish the state-of-the-art or the SOA.
All right, let's get started.
Okay, so as we discuss the Scientific Validity Report, it's really important to first discuss how to establish the state-of-the-art. So I'd like to start with a few basic definitions. And first off, just to define, what do we mean by scientific validity? According to IVDR Article 2, a scientific validity report should establish the association of an analyte with a clinical condition or a physiological state. So the basis for the SVR is, in effect, a detailed discussion of the analyte and the method of detection. That is essentially the same purpose as the state-of-the-art section as we recognize it in MDR. The IVDR guidance recommends that you should plan how to demonstrate that scientific validity of the analyte as early as possible in your development cycle. However, the SVR is a new requirement, so it's still not 100% sure what the expectations are to establish scientific validity of the IVD under evaluation. While we all await notified body feedback to clarify the level of detail expected, we should start by just relying on the extensive experience from the medical device world in order to develop a process and a framework to start to build off of. So how do you go about showing this association between the analyte and the medical condition? The SVR will help you explain this link, first by explaining the background of the clinical condition, the analyte, and the alternatives for diagnosing this condition. Are there multiple types of tests or technologies? What are the pros and cons of each type of IVD? These are all going to be critical components within a typical SOA section of the PER. So next, you will include current recommendations and guidelines related to clinical diagnostic testing for that medical condition or clinical state. This may also include relying on many of those SOA articles, but it could require additional ad hoc searches to help you identify statements that have been published from experts and organizations. This can be fairly straightforward if your analyte has a well-established association, or if it has a very long publication history. But suppose your IVD is designed to detect a novel analyte, or if there's just very limited evidence regarding your analyte in the published literature. So in this case, you may need to rethink the strategy necessary to establish scientific validity. And this is why it's recommended that you begin the scoping process early in your PER preparations, because limited data supporting scientific validity will significantly impact the quality of your PER as a whole. And then finally, you will be expected to define the competitive landscape. Competitors are different than alternatives, so keep that in mind. This is the section where you will be establishing safety and performance measures, and determining your acceptance criteria. And you'll be using data extracted from many articles retrieved through your competitor device search. Your goal is to compare your IVD to the competitive landscape of other IVDs to show that your IVD performs as intended and meets the pre-established acceptance criteria. Now, be prepared to document each of these searches in extreme detail, and you must present your results very clearly. The process of building the SVR template relies heavily on the results of your systematic literature reviews. But don't forget that this SVR must document this as a process, and the entire process must be objective and reproducible and very well documented. The notified body will expect to see that relevant articles were identified and screened and then appraised in a very transparent and a clearly defined manner. There are two different systematic literature reviews that we'll describe today, and we'll discuss in additional detail later in this presentation. So as I just mentioned, the SOA is going to be a critical component of your PER, but it's also vital to establishing scientific validity. So what do we mean by state-of-the-art? When you're describing something as state-of-the-art, you usually use that terminology to convey that something is a cutting-edge technology. And based on our experience with MDR, MedDev Rev 4 is a document that is really our best source document for guidance as to the methodology and the required content for a state-of-the-art section within the SVR. Manufacturers are expected to demonstrate a thorough clinical evaluation of the IVD in the context of what is generally acknowledged to reflect the most current best practice science. We need to establish scientific validity by documenting how your IVD fits into the scientific landscape. And we need to explain the circumstances and the diagnostic scenarios by which this IVD can be considered state-of-the-art. What that means is that you will be expected to present a systematic and organized process to continuously generate, collect, analyze, and assess clinical data pertaining to your IVD. The goal is to develop a reproducible and an unbiased assessment of the safety and performance of your IVD in the clinical landscape of all diagnostic options and alternatives. So that's where we're starting today. What do you really need to know before you can define state-of-the-art for an IVD? Now, we're focusing on the SVR today, but keep in mind, the goal of the PER is to effectively demonstrate scientific validity, clinical performance, and analytical performance of the IVD. And as you mentioned in the poll, there is a lot of concern with how do you integrate these three standalone approaches? How do they all fit into the larger scope of the PER? The SOA is that link that's going to connect each of these independent reports, largely by providing clarity about the purpose of your IVD, where it's positioned within the scientific landscape, and then the standards by which you will measure safety and performance. You're also expected to identify comparators that will be used to define best practice within that scientific competitive landscape. Our presentation today is focusing on the state-of-the-art, but that requires having a lot of these ducks in a row prior to beginning the state-of-the-art section. You need to have a thorough understanding of what is going to be contained within the APR and the CPR in order to really fully address those concepts within your state-of-the-art section. So before you begin to plan the SOA, have a firm vision about a few critical points. First off, strategy. Is your regulatory strategy finalized? And have you made decisions with respect to IVD groupings or families? Are you writing one PER or multiple PERs? This is a really critical decision to make early on, definitely before you begin writing your SVR. Next up, you need to make sure you have your feeder documents. So keep in mind that if you don't have all of the necessary inputs for your PER, that could delay the full development of your SOA. Presumably, many of you are at the stage where you're still composing many of these supporting reports, including the SVR. But do you have all of the inputs that you're going to need to prepare these documents? And are they in alignment? Have you completed your systematic literature reviews? And do you have the data? you need to see if your IFU has been finalized and if product inserts are available for review. That's part of our early steps in our scoping for identifying the current status of a project, is to consult those IFUs and product inserts very early on. The APR will be documenting the analytical performance, and that may include data obtained through in-house validation experiments, as well as any reports on the IVD that have been published in scientific literature. So this is where you will need to have a thorough understanding of scientific standards and guidelines to explain, how do we define sensitivity and specificity? And how does your IVD compare to established clinical practice standards and guidelines? The CPR is going to then present clinical data that's been identified through a systematic literature review. This may include safety data in terms of risk for incorrect test results, and it may also include data that presents on the performance of the device in a clinical setting. So a thorough knowledge of your patient populations, potential indications for your test, comparators, and the outcomes by which these tests are measured is necessary to really conduct an unbiased assessment of safety and performance in a clinical setting. Which leads us to the SVR. The SVR should clearly present the scientific landscape by explaining the science behind the technology and define what constitutes state-of-the-art in this setting. So that will require an understanding of the safety and performance objectives that will be used to develop your APR and your CPR. If these objectives are not supported within the state-of-the-art section in the SVR, your reports may start to feel disjointed rather than independent yet complementary pieces that they're intended to be. So now let's discuss the systematic literature review process and how you actually go about acquiring published data to support your state-of-the-art. Additional details regarding the systematic literature review process have been discussed in detail during previous Criterion Edge webinars. So I encourage you to check out our website for some of those recordings. In today's webinar, we will not be discussing the process in significant detail, but it's necessary to have an understanding of the purpose of the SLR in order to discuss how to establish the state-of-the-art. So we should start by discussing systematic literature review in the context of your SVR. At its heart, the purpose of a systematic literature review is to ensure that you have identified relevant scientific data through an unbiased and a methodologically sound process. This is a good time to point out that this is not optional. The systematic literature review is long considered the gold standard for evidence-based research, as most of you are aware. But that being said, while it's mandated as best practice, systematic literature review can also be extremely daunting. It's not just a PubMed search or a Google search. So as we think of the different examples, the examples that we're going to present at the very end of this webinar will kind of walk you through the overall process of how you define the steps involved in systematic literature review. The first systematic literature review you'll perform will help you identify articles and background that's relevant to the state-of-the-art section of the SVR. So defining the questions requires you to identify your PICO terms. And that implies that you have a thorough knowledge of your population, the indications, comparators, and outcomes. So with the help of a qualified reference librarian, you need to design a search that uses proper mesh terminology. And you should have a robust search platform that allows you to search simultaneously across multiple databases. That strategy will save you a lot of time and energy, and it's critical to the next stage, which is screening. Because every article you identify in your systematic literature review search must be screened for relevance based on your predefined inclusion and exclusion criteria. So if you have a poorly designed search, you could easily collect thousands of articles that have been identified. And what that means is that you could spend a significant amount of unnecessary time and delays during the screening step of this process. By sifting through the irrelevant or unrelated articles that are retrieved through poorly designed search terminology. The data extraction is the stage in the process in which you've identified the articles that contain specific data relevant to your performance criteria. And in the case of many IVDs, their performance criteria may encompass data such as sensitivity or specificity, false positives or false negatives, or limit of detection. And then in the end, you must present the data that's been extracted, but you're also expected to report on the process that we undertook to arrive at this data. So remember, we are looking to prove the association of an analyte with a clinical condition or a physiological state. So have you really given much thought to the importance and the impact of having a solid IVDR compliant template? Most people focus on that PER template, but it's just as important to develop a solid template for your SVR, APR, CPR, and your PMS, PMCF plans. So think of your templates as a roadmap. If you sat down and read just the template, each standalone report should make sense and it should flow logically. If you've designed appropriate templates, your PER should start at the beginning by setting the stage with the state of the art, and your story flows from there. The PER should culminate with your conclusion that yes, indeed, this PER has demonstrated that your IVD is safe, it performs as intended, and may be considered state of the art, which means it meets the IVDR requirements for safety and performance. So now we're going to take a look at some of the critical components within a typical SVR template and really look at how the SOA section fits into that. That being said, the template will certainly vary, and it will need to be customized based on the unique needs of your device. But generally speaking, the approach to establishing the SOA is essentially the same as in the MDR world. IVD Article 56 describes the state of the art in medicine in relation to the performance evaluation concept for demonstrating scientific validity, analytical performance, and clinical performance. So the data and the conclusions constitute the clinical evidence by which you will demonstrate scientifically that you can achieve the intended clinical benefit. So in this case, we will conduct the first systematic literature review to support the SOA section because it is such a critical section. This search will include broader search terminologies because the articles that you retrieve will provide the data relevant to the medical condition and your patient population. In terms of the alternatives, this would also include a discussion of other ways of making this clinical diagnosis, but it would need to include a discussion of the evolution of clinical testing recommendations. Is there a specific diagnostic algorithm that's recommended? If so, that should be discussed in detail to establish how your IVD compares to alternative testing modalities. The risks and the benefits should also be presented in terms of the technology, not the patient. So the clinical risks and benefits will be discussed in your CPR. The SVR should instead focus on the potential risks and benefits in terms of the ability of that test to provide an accurate, sensitive, and timely result. Now, you've probably noticed that SLR appears twice in our outline. During your PER writing process, you will be conducting multiple systematic literature reviews. But that also means that the entire process of defining and searching and screening and extracting and reporting will be repeated in the second half of your SVR. So we're going to focus on this section in additional webinars that help to explain that whole process of systematic literature review in detail and to distinguish between the purpose and the scope of the SOA search and the competitor device search. But ultimately, your PER should help you to combine that information from each of these searches to tell a single cohesive story. So as you're beginning to develop your template, ask yourself if your SVR includes all IVDR required elements. A great advantage would be if you had a template that's already been pressure tested by having gone through a notified body review. But IVDR is new to everyone, so it's really just hypothetical whether or not your template will pass notified body scrutiny. Having experience with MDR and CER preparations will be extremely helpful as you develop these templates. As you go along the process of developing these templates, ask yourself if it flows. Is it logical, and does it tell the story of your IVD? But on the flip side, relying on a template that's untested or disorganized can create downstream delays and cost overruns to fix a poorly written PER. And worst of all, it could delay the IVDR approval for critical devices within your product portfolio. So we definitely suggest that you take the time at the beginning of your process to ensure that the systematic literature review is conducted properly and that your SVR template is well organized. And that will help you ensure that you have established the state of the art for not only your SVR, but also the APR and the CPR. Some critical considerations to take into account: determining those comprehensive PICO terms, your inclusion-exclusion criteria, their performance criteria, and having a solid list of your competitors. All of those decisions must be made early in the SVR planning process before the writing can even begin. So the scientific validity report should show that the analyte correlates with a clinical condition. And I thought it might be helpful to illustrate what this might look like in a real-world scenario. So let's use an example of a common clinical diagnostic test, the C. difficile test. C. diff is a gram-positive, spore-forming, toxin-producing bacterium, and it's a major cause of antibiotic-associated diarrhea. So suppose your IVD is a PCR-based C. diff test. The C. difficile bacterium is your target, but what is the analyte that your test is designed to detect? In this case, your analyte is the gene that your test is amplifying by PCR. Many tests may involve more than one gene. So in this case here, you may have a test that targets the toxin A loci or the toxin B loci, or possibly both. So be prepared to justify not only your choice of gene targets, but also the number of analytes or the number of targets you've selected with respect to scientific validity. As you write the SOA section, the articles you retrieved in the SOA search provide the data relevant to the medical condition and the patient population. Clinical presentations associated with C. diff infection can range from an asymptomatic colonization to severe disease. So in this case, in this example, the medical condition is symptomatic C. diff infection. So your SVR should explain how your test is able to avoid detecting non-toxigenic asymptomatic strains, because that's going to speak to the clinical benefits of the assay. It doesn't help you to have significant numbers of false positive results if someone has one of these non-toxigenic strains. It's expected that many populations would test positive in this reaction, but you're specifically targeting someone who has one of these toxigenic strains. So you need to be able to show that your test is designed in such a way to distinguish between those two scenarios. And in terms of alternatives, that will include a discussion of reference tests. And in this case, we have toxigenic culture. We have a cytotoxicity assay that is sort of the oldest way of doing it. We have various enzyme immunoassays, a GDH and a toxin immunoassay, and then we have nucleic acid amplification tests. Each of these have benefits and differences in terms of their clinical utility. So be prepared as you're presenting your alternative sections to discuss this. If there is a specific diagnostic algorithm that's been recommended as part of routine C. diff diagnosis, you should discuss that in detail to establish where a nucleic acid test compares to alternative testing modalities. Are these tests used in combination or in a particular sequence? The risks and benefits should also be presented in terms of the technology, not the patient. So the clinical risks and benefits will be discussed in that CPR. So you're going to focus on the ability of the test to provide an accurate, sensitive, and a timely result. And that's going to help you with designing the overall approach in terms of your risks and benefits for false positives and false negatives. So to summarize, you would want to describe the C. diff infection and the clinical burden and the course of infection. That's your medical condition and background. Then consider how your test detects C. diff. In this scenario, you're amplifying a specific gene target through PCR. You should include a justification of your choice of gene targets. But then consider, do all C. diff tests detect the same target that your IVD does? That gets to the question of how have you established your competitive landscape? If you have multiple gene targets, how do you ensure sensitivity and specificity of your assay? And is your approach able to distinguish between those asymptomatic and symptomatic infections? Also consider what types of samples can be used, because that can affect the data that you're collecting as part of your competitive landscape, which will ultimately affect your acceptance criteria. In this case, you can collect test samples from fresh samples, frozen samples, or swabs, and they will likely provide differing levels of performance. So that can be included within your alternative section, but the articles that you've retrieved during your state-of-the-art search will help you identify whether other alternative or competitor C. diff tests work under the same premise using similar technologies. And that's going to help you support your choice of competitors that you're using to establish the landscape. And finally, you want to discuss the risks of the tests in terms of false positives and false negatives. Are there acceptable ranges for sensitivity and specificity for a C. diff diagnosis? Are there benefits of PCR testing over the cultures that establish that this is a scientifically valid approach? All of this must be covered as part of your SVR, but this is also why it's so critical to plan the outline to ensure that your discussion of the association is comprehensive and well organized. All of this background must be established before you can begin to assess what can be considered state-of-the-art. So to recap, we're hoping that by the end of this session, you have a better understanding of how to assess your own SVR project for any unanticipated roadblocks or delays. Are you ready? Do you have these feeder documents prepared, and have critical decisions been made prior to beginning your writing process? We hope you better understand what inputs will have the greatest impact on the SOA. And then ultimately, how are the SOA and the SVR related? We hope we've given you a better appreciation for why systematic literature review is a critical tool in this process, as well as what information should be included within your SVR template. And finally, I hope that you all have a better appreciation for how indecision or limited supporting documents can negatively impact not only the SOA, but also your SVR and ultimately the quality and the timelines for your PER as a whole.
Thank you, Sarah. We have about 25 minutes now to answer some of your questions, and joining us is President Lori Mitchell. Hi, Lori, how are you?
Had to unmute myself. I am great, and thank you, Sarah, for a great presentation. So again, thanks for your engagement, and Sarah is a great expert on this, and we look forward to answering your questions.
So with that, let's get to our first question here. So our first question is really, how important is the state-of-the-art in regards to the Scientific Validity Report?
Honestly, it's probably the most important piece of the setting your PER in the right direction. If you have sort of gone through the SOA and it's incomplete, or if it's not very well documented, it can completely derail even the best SVR report. They are going to expect, the notified body will expect to see that this whole process has been very well documented, because this is ultimately the data that you're collecting to set the performance, to set the acceptance criteria. So you need to show that the standards that you've identified for performance are what are considered to be the state of the art by experts in the field. And then you need to show that you didn't just sort of selectively choose certain studies to include and eliminate other studies because maybe they didn't support the efficacy of your device. So you really do need to make sure that this is a very well documented, very well organized process to set your SVR in the right direction. It's going to be a very critical component of the final PER report.
And I can jump in and say I 100% concur with that. In our experience with medical device companies over the past 5 years, The state-of-the-art has really emerged as, I think, as Sarah said, the foundational section of an MDR CER. It's foundational. It establishes your subject device, the device under evaluation, your subject device within the competitive landscape as state-of-the-art within your competitive landscape. So I like to say the state-of-the-art section puts your device in space and time. It locates it in space and time with its competitors, with what it does, with what it doesn't do, and then you go forward in your analysis having established those facts. So it's a great question. Thank you for asking it, whoever asked it, because it's foundational. We're glad to talk a little bit more about it. So Sarah, thank you.
All right, let's get to our next question here. What should we cover in the state-of-the-art if we develop a novel lay use test and there are no similar tests on the market?
This can be a challenge. We've encountered this with several clients where they, early in the process, we ask them for a list of alternatives and a list of competitors, and they'll say, we don't have a competitor. So that makes the SOA section even more critical. So if you are using a lay test, and I'm assuming that means sort of a point of care or a test that would not be a laboratory-based test, you're relying then on the only data that you're going to be able to collect if there are no other lay tests or point of care tests on the market, you're relying heavily on the alternatives. So you will still be collecting data and you'll still be expected to develop a range, but you will want to make sure that you take the time to explain where this type of technology fits in the landscape as a whole. So I would collect the data from the alternatives that you've identified and use that to explain sort of where this particular test is going to fit. It may not have the same sensitivity or specificity. It may not meet those acceptance criteria that you've established from a laboratory-based test. But if this test can provide immediate results in the field, that is still part of your risk-benefit analysis, and it still can be considered as being state-of-the-art with that respect. So you can't say I don't have any competitors and therefore I have no data. That's not going to be acceptable to the notified body. What you do need to do is establish the alternatives and rely on them more heavily and rely on clinical practice guidelines when you're dealing with a novel analyte. That burden for establishing scientific validity can be a little bit higher because you need to show how this test and how the technology compares to existing technologies.
So Sarah, I'm going to jump in and ask you to speak just another minute more about the importance of justification, providing a clear justification to the notified body that this is your approach and this is why, and this is how we're going to deal with our problem.
Yes, so we have within the templates that we've developed for the SVR and within the SOA section, we very clearly describe the decisions that were made early in the process with regards to the PICO terms, our patient population, and all of the different alternatives that are out there. If you note that there are no direct competitors, you should be able to establish what are the current methods for making this particular diagnosis. And then you may choose performance measures, things like time or the limit of detection or the time to results. That could be a way that you could show, no, we're not as sensitive, but the other alternative tests require overnight cultures, whereas this test can provide a result in minutes. But you would also be expected to relate that information back to the cutoff levels and the limits of detection that have been established within your APR. So it's all about connecting these reports and making it feel cohesive. So as you're developing the template for your SOA, justify your choice of cutoff values, justify your choice of performance measures, and justify your overall approach to how you designed and identified the relevant PICO terms and the relevant search terminology. That will help the notified body follow your train of thought and not get lost by saying, well, you've established that you're this type of technology, and yet all of the data you've presented is on this type of technology. If you've explained the limitations in the data, that will help them understand your train of thought.
Excellent.
All right, thank you, Sarah. Jeanette is asking, when establishing the competitive landscape, what are you looking at when you want to look at safety and performance? For instance, is this approved IVDs or research use only?
I typically start with your closest competitors. So if you can identify approved IVDs where they've already met the burden in theory, that's always more helpful. We often, as we're determining our competitors, we may start by consulting the marketing group within our companies, and they can kind of give us a sense of, if someone was trying to choose between different assays, who are our direct competitors from a marketing perspective. Some of those may be research use only, but many of them, if they're going to have relevant clinical performance data, they would be one of these approved IVDs. So that would be my hesitance towards identifying research use only, given that you may lack the clinical data that you're required to present in order to establish the safety and performance. So the approved IVDs will likely provide more hits and more data as you are doing these searches.
All right, thank you, Sarah. Our next question is, how do you search for information regarding state-of-the-art?
In many cases, this is part of that early conversation with the client that we have as part of our scoping. You want to cast a relatively wide net to capture enough information on the background and potentially any patient populations. But really, we tend to start with the IFU. We tend to start with the intended purpose statement or the product inserts. Those are going to give you a sense of how the terminology may have changed, what are specific terms, what are terms that you should include or exclude. If you have like something like the C. diff, the name of C. diff has changed over the years. So as you're developing your search terminology for the SOA, you would want to make sure you included those other alternative names just based on how it's described in clinical literature. But you also want to hopefully capture some of those clinical practice guidelines and recommendations, so you can include terminology within your mesh terms that is going to hopefully capture those clinical recommendations as well. There may be some gaps in the information that you identify, but often you can use citation mining. So if you identify a solid review article, you can look to some of the information or the articles cited within that meta-analysis or review to supplement the SOA. But ultimately, if you're identifying data sets for the alternatives, it's expected that you're going to screen this with the same level of detail and the same care that you would your competitor search. So you want to be targeted, but you also don't want, you want to make sure that you've found that balance between being broad enough that you capture all relevant information, but not so broad that you have to then screen thousands of articles.
All right, our next question comes from Ritu. What if you are using an analyte that no one else uses?
That's going to be the situation where the SVR is going to be critically important. In this case, like let's say you have a drug test and that most drug tests use one particular metabolite of the drug, or most of them use a parent compound, and you've identified that there's actually a metabolite that exists that is a better target for accurate drug testing. If you can justify through your APR that this analyte does exist and that you're able to detect it, you need to be able to use the scientific validity of that analyte to justify that this is also a valid approach to making this diagnosis. So that can come up too when we have certain tests where, let's say up until now, we've relied on culturing these microbes, and now there's a PCR-based test. Your analyte is now a gene in that particular microbe. So as you're justifying your choice of that PCR target, that analyte, you have to show what is any potential for cross-reactivity. Are there any potential interferences with that analyte may result in a false positive or a false negative? How specific is that analyte to the clinical condition that you've identified? And are there ways in which that analyte might not be the best target for that particular diagnosis? So you'll be expected that you may have to go into a little bit more detail and a little bit more of the biochemical backstory of your analyte within the SVR to really be able to justify where it sits within the diagnostic landscape.
Our next question is coming from an audience member. Is an SVR required for Class A or general equipment IVDs that is not linked to any specific analyte or condition?
This is going to be one of those sort of gray areas that I think is going to come up in terms of what's required and what's not. I would say that the approach that we would take is that we would use the SVR may not be quite as detailed. You're not going to necessarily, if you're using like a wash solution or something like that, you would not necessarily need to go through all of the same level of competitive landscape as you would. But you would still be expected to explain the scientific validity of that particular step in the process. So I think it would definitely depend on your particular test. But the template and what's included within the SVR will vary depending on the class of your IVD.
I think this is a great example of TBD, to be determined. We're still early in the process, and our clients and our IVD clients and what we hear on the ground with regards to the notified body is that this is still being discussed, is like, how far do we go with these Class A devices? How much is required to push to the notified body? So, I think it's our opinion right now, but I think none of us know for sure exactly. And I think more fine tuning for Class A devices with regards to the IVD requirements is yet to come.
All right, thank you. Here's our next question. How much information would be considered sufficient in SVR?
We get this question a lot. I think everyone's looking for a bare minimum, like what is the minimum number of points I need to pass this test from the notified body? What it really comes down to is there isn't a set number. You are expected to put in your due diligence and really show and document that you have tried your best to collect as much information that's out there as possible. So I think sufficiency is really justified or proven by the approach that you take and the search methodology that you've employed. If you can show that you have a test that maybe it's not very common, or this is a test that's been around for years. So I searched the past 5 years and there were only 5 articles that used this test. That might look like not that much information, but if you can then state, I found previous meta-analyses that show that this test was used widely and reported on widely in historical records. And, like going back 20 years, this test has been widely used. You can then pull in information from previous published meta-analyses, clinical recommendations and guidelines, all of that sort of information to help you justify and support your acceptance criteria that you've developed. It's going to come down to sort of how common is your test? How often is it reported in the literature? Are there other alternatives or competitors that are more likely to be published? But yeah, it can be challenging if you have a particular device or a type of test that just is not very well published on. You will definitely want to pull from some of those alternatives or clinical practice guidelines.
So Sarah, you gave some great solutions there, or suggestions to navigate this conundrum, which is what if our device, there isn't much data on our device. This is a shared problem over in the medical device world as well. We would expect the notified bodies would approach the solutions under IVDR similar to the way they approach it under MDR. That would be our expectation anyway. And we can say that data sufficiency is almost the top question that we're asked, or that we grapple with as a team, with regards to when we're writing up a CER and how you'll be doing it for your PER or an SPR or a CPR. And so appreciate, Sarah, you kind of weighing in on this, because this is a top question we get. One thing that I'd like to finish with this is that the notified bodies, they will take reason and justification. In other words, if you well document in your reports, SVR, the PER itself, that you looked everywhere, that you did these things, this is down to your documentation of your methodology for searching for gray literature searching, as Sarah just described, or systematic literature review searching, or explanations or justification why there isn't very much data, and that you've looked under all these rocks and you've done your due diligence. The notified body, in our experience under MDR, takes that into consideration. If you well document it within your reports, that you've looked everywhere, you've done these things, it's methodologically robust, they will take that lack of data into account. In other words, you haven't left any stone unturned. The data are the data. And that has been our experience as well in the MDR world. So those are my additional thoughts.
All right, thanks, Lori. Here's our next question. Do we need to differentiate SOA in the SVR versus the PER?
Really, the SOA section is what it is. It is establishing the state of the art. It makes the most sense for it to live within the scientific validity report, because you're establishing the validity of the approach itself. You're establishing that the performance criteria you've selected are all valid. So when you are writing the PER as a whole, it's not just going to be a template where you copy and paste all three SVR, APR, CPR, you just staple them in and call it done. Think of it like writing a graduate thesis. If you tried to turn in a 1-paragraph introduction and a 1-paragraph conclusion with your 3 papers you published in between, that's not a thesis. You need to connect those 3 standalone reports. It's not just importing the entirety of the report verbatim into the PER document. So I would expect that you will pull the SOA section from the SVR report into the PER, but there may be additional background information contained within the CPR and the APR that may require you to pull in some additional information to supplement the SOA section. But generally, the SOA within the SVR should be fairly complete in terms of setting the landscape for these other complementary reports. But it's just to warn you all, it's not going to be just a copy and paste kind of approach. It is going to require more of a synthesis of these 3 documents to make the PER flow well.
Okay, great. Our next question comes from an audience member. Is the SLR applicable to background information concerning the analyte's function, or is this specifically for the clinical relevance of the analyte in current devices?
I typically approach this to present the analyte's function as a general rule because I do think that it helps to understand how and where and why that analyte would be expected to be identified. Like, let's say you have an analyte where you're detecting, like in COVID testing, if your analyte is the spike protein, you would want to explain the general background information about the immunogenicity of the spike protein, how, what's its structure, what is its function, how often is it mutated, that sort of thing. So you want to give enough information about the target to where when you're trying to explain the link between the target and that function, the clinical relevance of that target, it's established, you already have an understanding of what the target is, if that makes sense. It's you need to show that analyte, that detecting the spike protein is linked to COVID infection, but you can't just stop and say that it's just the spike protein is linked to it. It would be expected that, and it's going to help you understand sort of potential interference or contamination or false positives, false negatives by understanding the background on the function of the analyte itself. Maybe there are situations in which it's not as easily detectable, and that's where I include more general background information on the choice, on the target itself, prior to showing that clinical relevance.
All right, thank you so much, Sarah and Lori. That is all the time that we have for today. Thank you everyone for attending today and for your wonderful questions. We hope this presentation helps you get one step closer to IVDR readiness. Take care, and I hope to see you at our next webinar.